So we're here at the first part of the central nervous system, but we're going to finish up neural tissue because there's a lot to go over. And do remember that you have the quiz today. All right. So we didn't get to this last time, right? I don't think so. Okay. We got through, we have a neuron, it's great. You've got this resting membrane potential that you set up, right? And based on events that happen, you can change that potential. You can make it send a signal along its length, right? You can make it basically zap electricity the whole way down. Pretty cool? Well, that's great, but just having a neuron zapping by itself isn't super exciting. The nervous system works because information can jump from one neuron to the next. Back in the day, it used to be people were trying to figure out what was sending the signals in the nervous system, right? And so there were two camps. They were called the Sparks and Soup camp. The Sparks camp were like, look, there's a whole bunch of electricity, there's a whole bunch of membrane potential changes that are happening, right? There's like, the brain looks very electrical. So obviously the signals are being sent electrically, right? Which seems reasonable. The other camp was, whatever. So yes, electricity, cool, whatever. It's something that happens because of, physics. But it's gotta be chemicals. These people were called the soup people because there's like little chemicals floating in the goo. Like the signals being sent via chemicals, right? Spoiler, they both were right, or equally wrong. But I'm pretty sure, I'm not actually positive, but apocryphally there's at least one duel fought about this, of like, you believe this, and I believe that, so I will, challenge you to duel. Which, I like where we've come with science, and I'm glad we don't threaten violence quite so much. But part of me feels like it would spice up meetings if, like, you don't like somebody's research and you threaten them to sword fight or something. Anyways, all that's to say, the part where things actually start happening is at the synapse. So, this is what we've got going on here. We've got, let me see if I can show you right. See that neuron there? That big yellow one, right? And it's got an axon heading out for who knows where. There are other axons from other neurons that are now touching this cell. So that can happen, an axon from something else can touch the dendrites. Dendrites are great at receiving information. The axon can touch the actual cell body, but these axons from other things, you can see it here. This is the neuron. So neuron A, neuron B is coming and touching it. This is an axon, has these little ends, they're called axon terminals, or terminal boutons. They're touching it, right? They're not actually technically touching it, there's like a tiny, tiny gap between them, but they're right next to it. We're good? And now, what's going to happen is the first neuron is going to send a signal, boom, via the action potential down this axon. Yay! It's going to get to the terminal, to the axon terminal, to the end of it, and it's going to then go from being an electrical signal to being a chemical signal. Okay? So, if we have this picture going on here, the system is set up to handle electricity, right? Sending electrical signals is way faster than sending chemical signals, because except chemical signal, you basically have a bag of chemicals, you pull it open, the chemicals spill out, and then you wait for diffusion to ooze them over someplace, and then you receive the signal, right? This is basically the endocrine system, right? You dump chemicals out into the system, you wait for things to happen, right? Whereas an electrical signal, you start, boom, you send it. It's really fast, right? So the really fast thing is good. right? So you can respond if you step on, I don't know, something sharp and pokey, you respond really quickly. You don't have to wait for the chemicals to diffuse. Any problems, though, with having an entirely electrical system? If everything was just electrical, no chemicals. Actually, better question. With action potentials, can you vary the... You can turn them on, right? You can turn them off. Right? That's what you get to do. It gets you a lot. But can you have more than, say, 2 chemicals you could think of? Presumably, right? You could think of a bazillion different ones. You can have a bunch of neurotransmitters. The way the classic synapse gets set up is the signal inside the neuron is going to be sent via that electrical change, right? The signal between one neuron and the next is going to be sent chemically. And this sets it up so that the whole system's really, really fast, but you also could have more than just on-off being said. You could be sending acetylcholine, you could be sending dopamine, you could be sending serotonin, you could be sending basically anything that's a neurotransmitter, you could be sending that. So now you have a really fast system that can say lots of different things. Are we comfortable with this idea? The nice part is, though, that the chemical part, the part that needs diffusion, only has to cross a tiny, tiny space, because between where this axon ends and the signal's received over here, that space is tiny. So diffusion's nearly instantaneous. We're good? Great. So this is called a chemical synapse. They're the most common type of synapse. So synapse is the axon terminal of the first one, the one that's sending the signal, and then the receptor region on the other side, and the little tiny space between that's called the synaptic cleft. So typically, so the axon terminal, that one belongs to what's called the presynaptic neuron, because that's the one that's happening first. And then the thing on the other side is called the postsynaptic region. If cell A is talking to cell B, this one's talking to this one. A is presynaptic, this one's postsynaptic. But B could go talk to another cell, and now B is presynaptic, C is postsynaptic, right? It's just saying who's sending it, who's receiving it. So far so good? So the signal's going to go from the presynaptic neuron, from the cell body, right, of the neuron. It's going to go electrical signal, change to chemical signal, jump that cleft, hit the other cell, typically another neuron, that could be a muscle or gland or something, hit that other cell, and change back, if you're in a neuron, back into the electrical signal. Okay? Here's this beautiful picture. Is that big enough? What we have going on here, we're zoomed really, really close in. Axon terminal, electrical signal comes down, phew, like you saw with muscles. That comes down. This action potential, when it gets here, you've seen this in muscles. You've seen this in muscles. Action potential gets here, opens voltage-gated calcium channels. Calcium comes in. That causes the vesicles to spill their contents into the cleft. This is exactly what you saw with, say, motor neurons releasing acetylcholine. But it doesn't have to be acetylcholine. It could be dopamine. It could be something else. Okay? Those neurotransmitters diffuse across the space. They hit ligand gate is that they hit chemically gated ion channels, right? And so this ligand then binds and opens up this channel, and so now ions can flow in. Let's say you've got something like you let sodium in. So if you were to let sodium in, you'd make this membrane way more positive, right? If you make the membrane positive enough, you could hit threshold. If you hit threshold, you'll send another action potential from this second one. Are we okay with that picture? The idea? Great. But what happens if instead of this chemical here binding to this channel, opening it up and having it open up a channel that lets sodium through, what happens if it opened up, say, a channel that only let potassium out. Or maybe it opened up a channel that let a negative ion in. If this lets a negative ion into this postsynaptic side, what happens? Yeah, it gets hyperpolarized, right? So now this postsynaptic neuron's less likely to fire an action potential. Okay? Signals don't have to just be go. You can have a go or a stop signal, right? You can have either way. And that actually leads us into the types of what are called postsynaptic potentials. So this is, you got the presynaptic neuron firing an action potential. That action potential gets to the end, to the terminal there, causes the release of neurotransmitter. The neurotransmitter has gone and bound to receptors on the postsynaptic side. And that's caused a change in membrane potential on the postsynaptic neuron. There are two types. There's excitatory postsynaptic potentials, they're called EPSPs, excitatory, or there's inhibitory, IPSPs, they're inhibitory. Just for the record, people often act as if a given neurotransmitter, I took out a whole bunch of neurotransmitter stuff because I got excited about not spending too long on things. There are a whole bunch of different neurotransmitters, but the neurotransmitters themselves don't by themselves mean excitatory or inhibitory. So like acetylcholine, in some situations in the body is excitatory, and in others is inhibitory. And just to be clear here, I think I used this example in neuroscience, so sorry, Tanner, if you've heard it before. Have you people ever been to like some restaurant for your birthday, right? And somebody you're with tells the waiter like, hey, it's their birthday. And then the waiters come out and they give you this kind of thing, like a candle and ice cream, and they sing and they clap, right? Okay. I think my dad would rather like break his arm than have this happen to him, right? It would just be like, oh my gosh, right? And then I have a buddy who, if she felt like she could get away with this, would do this every time she goes there, right? Because be like, yes, everyone's looking at me, and I get the candle and the cake, right? Okay. That actual event by itself, the candle and the ice cream, is neither excitatory nor inhibitory by itself. It's not positive or negative just by itself. It's how the person receiving it responds. Does that make sense? So there are some neurotransmitters that are almost always excitatory. Just like there are some experiences, finding $10, that's like always positive, right? There are some neurotransmitters that are almost always negative, or that are almost always inhibitory, right? Just like, I don't know, stubbing your toe is always just like bummer. But then there are also some that the experience itself, say, I don't know, getting a text, right? This could, depending on what the text is, could either be could be positive or negative for you. Are we clear on this? Great. So I'm going fast, but I think I have to. Are people following along? One single excitatory postsynaptic impulse isn't enough to trigger threshold and get an action potential to go. It's not enough. It's a little bit, it makes the membrane a little bit more excited, a little bit less polarized, but it's not enough to pull all the way up to threshold and have an action potential actually happen. But you can have more than one excitatory postsynaptic potential. You can either send, I'll just show you the picture. Here we go. So we've got this nice little axon here coming down to this neuron. It sends an excitatory postsynaptic potential. Well, it causes one here, right? You can see threshold right here. This action potential, as it comes across the thing, raises the membrane potential of this neuron but not to threshold. So bloop, raised a little bit, it gets reset. Bloop, it sends another signal, raise it a little bit, comes back down. I really like this picture. I don't know why. Of our little neuron hanging out in their rocking chair. It makes me happy. But the analogy, right, is if someone's like, panic, every few seconds, you don't really care because, like, whatever, they're not, you don't take it seriously. The deal with this, though, this excitatory postsynaptic potential, they can add together. So if there are two action potentials that are really close in time, or lots of action potentials really close in time coming down from this, the first excitatory postsynaptic potential raises the membrane potential slightly, right, but not enough. But the next one raises it a tiny bit more. And if you have them close enough together and they hit membrane that threshold potential, that next neuron does have the action potential. Do we see that? So you can do it like that by having them receive lots of excitatory signals close in time. You can also do this, where instead of having the same axon send lots of signals, you can have lots of different neurons send the same signal to the same neuron. It can converge. Right? It's called spatial summation. So the first one's called temporal summation. Right here. Well, I guess it's the second one. This is called temporal summation because it's being added together in time. It's really close together. This one is spatial summation because you've got two things at different places summing together on this neuron. So far, so good? Same deal. This one sends an excitatory signal. That one sends an excitatory signal. They add together, hit threshold, and you can fire an action potential. Inhibitory postsynaptic potentials also sum together. So if you have one input coming in and it's inhibitory, it brings it down a little bit. Another one coming in brings it down even farther. You can have those sum together. And you can have complicated patterns where you've got some signals coming in that are excitatory, some that are inhibitory, and the cell does interesting, the receiving neuron does interesting computations figuring out what this means. We're good? Questions about this? Concerns? Just for the record, most neurons are receiving thousands of other inputs, right? Like, it's not just one-to-one, even though this nice little picture acts as if there are only two. We're talking thousands. But as they sum together, the cell does computations and then response. Everyone's cool with this? This is called synaptic integration because you're putting together all the different synaptic potentials. Whew. Yes? Great. Okay. Neurotransmitters. The book goes into a lot of detail here. I'm going to operate under the assumption that people are dutifully reading the book, like, all the time. Don't tell me if that's not the case, but I'm going to behave as if you are. I hope you are. But your book goes into a lot of detail here. We're going to look at just kind of the basic categories. There are at least 50 different neurotransmitters, right? So these are just chemicals that are being used to send signals around the nervous system. Most neurons are going to make at least 2 different neurotransmitters, but we're going to ignore that. But basically, depending on how quickly the neuron is stimulated, it can release different neurotransmitters, which means neurons can do a lot of things. You know how computers operate with just signals that are zeros and ones? They do on-off, and because you have because you've organized it in such a way, your zeros and ones can do very, very complicated things. Imagine if you just exponentially increased your options. So you know how complicated computers are, they can do tons of computations. Now, in the nervous system, we can do on-off, but we can do on-off, and each of those on-offs can come in several different flavors. So you can have on-off this neurotransmitter, on-off that neurotransmitter. That's just mostly on. You can't really turn off. But you exponentially have more signals. The human brain is uber complicated, and it's beautiful. But these neurotransmitters, whole bunch, basically you classify them either by what they look like or what they do. When you're looking at what they do lots of things, but basically, well, let's just look at here. You can classify them by whether or not they're typically excitatory or typically inhibitory. So, are they going to depolarize things, or are they going to hyperpolarize things? Lots of words here, I realize. Something like GABA is almost always inhibitory. It almost always chills the receiving neuron out, so it's less likely to go. Sometimes I think people act as if inhibitory signals are bad, like, oh gosh, that's like putting a cramp in your thinking style, right? But talk to me for a minute what happens if your car suddenly doesn't have a brake system, like the brake line gets cut? That's good, right? Because you can go faster, nothing holds you back. No? Are we okay that having the brakes cut is a bad deal? You need the inhibitory signals going on as well, because otherwise bad things happen. So like GABA is usually inhibitory, glutamate is usually excitatory, but then some things like acetylcholine or norepinephrine, sometimes they're excitatory, sometimes they're inhibitory. So you can classify neurotransmitters by whether they're excitatory or inhibitory, but it's not a perfect classification. It's just kind of a general, let's see what they do. You can also classify them by, okay. We're still going for what they do, but this time, do they act directly on something, or do they have a long path for their action? Direct action neurotransmitters, they're something like acetylcholine. You saw this before, right? It goes through the synapse, comes out of the synapse, goes across the little cleft, hits a receptor, and that receptor, boom, opens an ion channel. And ions come in or they go out, but it's fast. These direct action ones, they're changing the membrane potential. Boom, they change the membrane potential. That's what they do. Indirect action ones, do I have pictures? I do have pictures. Let's go through pictures. Indirect action ones act on this long, beautiful second messenger process. And so the signals they're sending usually aren't things like fire, don't fire, or they're not quite so fast. They're more things like, you should make different proteins, neuron. You should grow this way, neuron. You should make less of that protein, neuron. That kind of thing. It's still a signal, it's still being sent. It's less immediately obvious than like, kazoom. So this would be some sort of, direct action, right? This neurotransmitter comes in, binds to this, immediately the membrane potential changes. If you have something like this, this is an indirect reaction. Neurotransmitter binds. No ion channels are involved right yet. A whole beautiful second messenger system. You end up sending a signal to the nucleus saying, you know what? I think you should maybe make more ion channels and put them in the membrane. Or maybe you should make less ion channels and take them back. You've heard about this before when you've heard about how like certain drugs, when you first take it, you feel really, really intensely, right? Like, whoa. And then over time, you have to take more drugs to feel the same way. The reason for that is because, amongst other things, you send indirect signals like this that cause more channels to be put on the membrane, right? So basically, the receiving neuron realizes, hey, I'm getting a ton of this chemical messenger. I don't know how to handle it all. I'm going to build more receptors to put outside to receive them. And then as you do, or less, depending on the kind, and as you do that, you've changed that neuron's ability to respond in the future. Do you see that? So indirect actions, super important. They're less immediately obvious, right? They take a while to act, but they're still really important. They happen all over the place. So far, so good. Okay. Briefly, and I realize I am just cramming like the whole nervous system. We have like 3 days for it, and then we have like a day and a half for the central nervous system, which I realize is not enough time. But the basic pictures. One neuron's nice. Lots of neurons are way cooler. Neural integration is just the idea that neurons play together as groups. Right? So one neuron is cool. When it's sending or receiving signals from another one, it's better. But when you have thousands or millions playing together, you can have some really complicated things happen. Don't memorize this, I just want to show you them. Neural processing works in lots of different ways. We'll see this. We're going to see this again when we talk about, say, reflexes. A very straightforward version of processing. It's called serial processing. It's where one signal chunks along an entire pathway to the end. It's something like this, where you have a stimulus, you step on the Lego or something, the signal is sent along this neuron, it's received by another neuron, right? This neuron sends a signal to another neuron or muscle or whatever. So this one sends it to the neuron here, sends it to the neuron, This neuron sends it to the motor unit, it sends it to the muscle. The muscle then responds, says, hey, don't step on the Lego, right? Serial processing meaning one thing happens, the next thing happens, the next thing happens, the next thing happens. Great. Most like complicated higher order functions aren't this straightforward. There's a lot of things going in, a lot of things coming out. Briefly, just to see if people get the reference. Have people seen this movie, Ratatouille? Okay. I feel like I need to watch more movies because I don't know what movies to reference for you all besides like Disney ones and Pixar because I assume everybody's just seen them. Anyways, so. Briefly, our little buddy here, and you can watch it, it's there. Our little buddy here takes a bite of like zucchini and then like 1 second later is thinking about feeling home and love and childhood and how it felt to be a small child and his kitchen back when he was 5, right? Okay? Which is not a direct response from like zucchini. Thoughts of home, right? Like something happened there, right? Like a lot of neural integration went down. Which like, you've had this happen too, right? You're doing something and it's not a one-to-one response of exactly what happened, right? It's not you step on something, you pull your foot back, right? This is called parallel processing. Information's traveling along lots of different pathways, lots of different circuits dealing simultaneously with the information. So signals come in, they split, they do a bunch of things, they come back. Higher level processing has this happen. And it shows up in lots of patterns, so something like this, right? So where one signal diverges and activates tons of things. You'd see something like this where one little neuron in the brain ends up telling thousands of muscle fibers to move because one neuron then the signal's amplified, and then you end up moving like a whole bunch of muscle fibers. You can also have converging circuits. So if you're thinking about so maybe, I don't know, the smell of cut glass, or if you hear a certain sound of a tractor, or if you see, I don't know, something, they all remind you of, I don't know, when you worked in the lawn care business, right? Like a whole bunch of different inputs all converge to the same output. That's really pretty common too. This one is my personal favorite. It's adorable. You don't have to be able to draw this. It's called a reverberating circuit. And this one is, so like back up here, this converging circuit, right? Makes sense, like lots of information feeding into one thing. This one feeds back on itself. So it's something where this neuron activates this neuron, which activates this neuron, which goes back and activates this neuron, right? Like it ends up looping, which is one of the few examples in the body of where you can actually get positive feedback loops. You can also have negative feedback here. You have positive feedback loops. And this sets up a lot of arrhythmic activity. So if you have this long enough and complicated enough, you can have something where, like, you wake up, and then a series of events happen in your brain, which is basically keeping track of time for you, more or less, right? Where it's like, do. Okay, in that case, like, this happens, so this happens, so I'm gonna release this chemical, release this chemical, release this chemical, release this chemical, do, back to, hey, I think it's night, we should go to sleep. And you set this cycle. It also is going to set up cycles for things like breathing, which hopefully is just this fairly repetitive once you've taken a breath. Your nervous system's just ready for you to take the next one, right? Just once you finish, you're back up. We're okay with this? These are really important. They're also really important for memory. These also are the tricky ones for epilepsy, because if things go wrong and it's not properly controlled, you can amplify this really, really quickly. So you can basically make one neuron yell loud, and then says, yell louder, and it just gets going out of control. I mean, you'll see that in seizures. There's also some different ones, which is basically apparently how, if you've ever known somebody who can do mental arithmetic quickly, they figured out how to use these ones, where you've got one input and one output, but a whole bunch of neurons are involved in the middle, right? So input in, I have some math problem, a whole bunch of neurons are going to work on solving it, and then we have one output. Is everybody okay? I hope whoever's outside is okay too, whoever loud sounds. Neural tissue, comfortable with it? Sweet. Neural tissue is amazing. But where it gets actually amazing is, now, brief transition here, is when you actually get it to be in globs. So today, we're going to very, 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 very briefly just look at the parts of the central nervous system. We're not going to get through all of them, and that's okay, because you will get out early to do your quiz. We're looking at parts of the central nervous system. If we have time, we're going to look at the cortex, central nervous system, brain, spinal cord. Okay? The spinal cord, well, okay. The brain is going to be divided into a couple different parts. It's got the cerebral hemispheres. That's the part that, like, if you look at it, you're like, brain, right? The hemispheres there. Covering, so the hemispheres, the two halves there. Also, if you do this with your fists and hold them together, that's about the size of your brain, which I feel like it should be bigger personally for me. I'm like, that's not that big. Probably about 3 pounds, just kind of there chilling, being amazing. The hemispheres, right, they're stuck kind of on the back, is what's called the cerebellum. Beautiful little brain there on the back. It's not a different brain, it's part of the same one. Covering the outside there of the cerebral hemispheres, you can call it's got this gray matter, it's called the cortex. Cortex means bark. Every anatomist has apparently called something the cortex. We have cortex in bone, we have cortex in the kidney, we have cortex in the brain. It's that outside covering. Yeah, you're going to see in the whole system, you're going to see gray matter and white matter. Gray matter, you're looking at the cell bodies of neurons. You're also looking at unmyelinated dendrites or unmyelinated axons. If you see white matter, you're basically seeing axons. And you'll see something like this pattern here. Bless you. So here's the brain, gray and white matter. You'll see a pattern like this. They're kind of backwards. In the spinal cord, you've got white matter on the outside, gray matter on the inside. And then in the brain, you've got white matter on the inside, gray matter mostly on the outside, but you also have gray matter blobs in the middle. Different nuclei in the middle. We're good? All this is showing here is that, okay, we've got cell bodies living here in the gray parts. I realize it's like pinkish on your slides. And the white parts, we've got these axons, the connections. Everyone's cool with this? That's the basic part. Have you noticed though, like here in this picture, there are holes? Right, there's like a hole here, there's a hole here, there's a hole here. Even in the spinal cord, there's a hole there in the middle, right? Is that bad? Is this just a terrible picture of like somebody with like mad cow disease or something? Is it supposed to have that hole right here? We're gonna look at another picture. Well, let's see. Do we have any in here? Let's see. Sure. Look at this picture. Ignore the colors on this. But do you see those spots where there's not white matter or gray matter? There's holes. You're supposed to have those. You like really have to have them. They're called ventricles. They're not like holes. They're not like empty, but they're these spaces that are filled with fluid. And they're going to help for a couple things. They're going to help, neural tissue is heavy and globby, and you want to support it. So you basically, amongst other things, you've got basically balloons full of fluid inside the brain to help keep it puffed up, so it doesn't collapse in on itself, right? So you've got fluid on the inside, you've got fluid on the outside, so it doesn't just mush. It also, cerebrospinal fluid helps bathe the brain and make it feel happy, make sure the ion concentrations are right, make sure gross stuff gets washed away. It's there. They're called ventricles. They're all over the place. They're in this cool shape. I don't have a good name for this shape, but they're all continuous with each other, and they're like this. If you've ever known a kid who's got hydrocephaly, have you seen this one before? Kids' heads get really, really big. The problem there is the ventricles, it's supposed to drain, cerebrospinal fluid's supposed to drain, but if there's a slight block in it, it can't drain as fast as it's being produced, and it'll puff up, puff up, puff up, and that's a real problem. What you do with it is you physically just put a drain in, and you can drain it to the kid's belly, and as they get older, the opening gets a little bit bigger. Okay, so that's the parts so far. The hemispheres are where most of this is at, actually. They're like 80% of the brain. And you've seen them. They've got bumps, and they've got little grooves. a bump, a little bump up. All right, so this is the brain, we look at here. A little bump up is called a gyri. So a gyrus. A gyrus is a bump up. Sulci are the grooves in as you go down, like the little valley things. And then if you have some really deep groove in, it's called a fissure. So you see that here, right? Bumps up, bumps down. That's helpful because you're trying to cram as much cortical space into your skull as possible. And so there are some animals that have smooth brains. Like mice have very smooth brains. So they've got their cerebral hemispheres there, you've got the brain, and it's just, the cortex is just there on top, it's smooth. Which is great. But you can only fit, like if you know your brain is, if your skull is this size, you can only fit so much flat cortex there. If you do the equivalent of crumpling it up, which is what you've done with something like this, you can fit a lot more actual brain into the same amount of space. Do you see that? So congratulations, you and dolphins and elephants have very lumpy brains, and that's good. Every single, and I kid you not, every single bump on there has been named. Every single bump up and every single bump down. They've all been named. And while people have slightly different, like the size of this bump and the size of that bump, it's the same between person A and person B and person C. You're expecting this major fissure down here. You're expecting this major groove down here. You're expecting this groove right here. Like every single thing. There are slight variations, but it's like there's slight variations in, say, how long people's fingers are. You're not expecting people to suddenly have like 3 arms. Brains are organized in very similar ways, and now it bugs me deeply when I see, it doesn't bug me deeply, it bugs me a little bit, when I see brains drawn in popular media, and they mess them up. They just kind of like put random lines in there, and it's a problem. While we're on feeling frustrated about things you shouldn't feel frustrated about, I was thinking about this morning, driving here. Coming off the highway, I come through here, and there's a little sign that says, like, snow route. Okay with this? It's got a snowflake on it. It has an 8-sided snowflake on it. And Frozen 2 also did 8-sided snowflakes. And they're not 8-sided, they come in 6 sides. I'm okay with the magical part, but good grief, just get the snowflake right. Anyway, sorry. Doesn't anybody else know? Okay, nope, you never thought of it. You're like, I have other things with my life. Spend my emotional energy other places. But like, how hard? Come on. Sorry. Thank you for humoring me. Okay. Do other people have things that they care about like this, or are you just like, I've got enough stress in my life? Okay.
Certain like grammar errors.
You're like, just get it right. Okay, yeah. Sorry, if I do it and it annoys you, I'm sorry. Okay, sorry. On with that, though, don't worry about memorizing all the little names of all the bumps, because you've got other things to do. But there are 5 big chunks of the brain. So far, there are lobes of each hemisphere. You should probably know these ones. So if you'd like to follow along, you can do that, like with your head. They're basically named for the cranial bones that are on top of them. So you've got the frontal lobe, which is this one right here, color-coded blue. You've got the parietal lobe back here. The temporal lobes are there, the sides, kind of by your ears. The occipital lobe is there in the very back. And then, if you go in right along here, there's a lot of gray matter in there. It counts as its own lobe of the brain. It's called the insula, because it's inside. It's what you see in green right here. All right? So frontal's pretty easy. Temporal's there on the sides, parietal, occipital, and then insula's more inside. These you should know what these are. Just a sec. Let's see how far we're going to get. I don't know if we're going to get to the sing-along video about lobes of the brain. We'll see. It's not a sing-along video, it's like a music video, actually. Yeah, we might not get there. What we will get to is these different lobes do different things. So, briefly, have anybody ever heard of the idea of phrenology? It was this idea back in the 1800s, bunk, by the way, that like if you felt the bumps on people's skulls, you could be like, by feeling this bump, I know you are a criminal, or by feeling this bump, I know that you are super musically talented. Heard of this? Good, okay. It was a crackpot idea, it's not a thing. That being said, you can't tell like anything by feeling bumps on people's skulls. So that was one idea, and people then were like, I don't think it's true. So then the idea scientists were thinking about was like, okay, you got this glob of brains, you have a lot of it, it kind of maybe behaves sort of like a muscle, so if you have a lot of it, you can do a lot of things. If you damage or hurt any part of it, you just kind of damage or hurt things in general, but it's all basically doing the same stuff. So it doesn't really matter which part gets hurt, it's just a little bit hurt, a little bit impaired, a lot hurt, a lot impaired, right? Where we are now is we actually know there are specific regions of the brain that do specific things. So you don't have one specific spot in your brain that's like, will you be good at music? Yes, no, click here, right? But there are parts of the brain that do, say, hearing. And there are parts of the brain that do things like tone of voice, when you have to go up and down. So you can kind of imagine this more like if you're thinking of an orchestra almost, we've got like the brass section, right? They can do similar things, slightly different, but they're going to be somewhat different than, say, the woodwind section, right? Typically, if one very tiny specific part of the brain is damaged, typically the neighboring areas try to cover for it, and usually it can. There are a few portions that if it goes, that functionality is just lost. Like if the one cymbal player in your musical ensemble gets struck by lightning, nobody else has a cymbal. Right? Like, sorry. But if one violinist goes, it's sad, but say the violas can probably cover for them a little bit. We're okay with that? One of the ways you check this is you pop people into brain scanners and you tell them to do things and then you watch their brains and see what happens. It's more steps than that. But then you get these pictures like this and you're like, oh, this person is looking at visual information because, well, as this person is looking at visual information, this part of the brain lights up. And they're not worried about listening to anything, but they're worried about looking at things. One more set of concepts. Well, let's see, how far do you want to go? How fried are people, by the way? I realize you have a million quizzes and stuff. Is it just calming to hear people yap about things that you'll then study later? Are we super into it right now? I'm just going to keep chugging away because I find it interesting. And I don't read facial expressions, so, I'm just going to go for it. Things, but ask questions. Okay. The cortex, so the cerebral cortex. The cortex is going to do. If you've got brain space for one slide, this is probably the one for today, but they're all important. The cortex is going to do a couple things. Okay. So, in the cortex, you're going to have areas that are controlling voluntary movement. They're not physically making the muscle move, right? Those are going to be motor neurons someplace else. But motor areas are the ones that are going to be like, I have a plan for juggling or whatever, right? It's going to be controlling, like, the neurons themselves, as they act, they're going to, through a series of events, bless you, cause motor movements to happen. Fair enough? Okay. There are going to be sensory areas. So motor areas are going to be sending those signals out. Sensory areas where sensory information is being fed through the body up there, and it gets to the sensory area, and the sensory area is like, oh, I can read this. Okay? And then most of the brain is neither motor areas nor sensory areas. It's what's called association areas, and it's putting things together. So maybe an association area listens to sensory information from taste, and listens to sensory information from touch, and is listening to sensory information from vision. And it's putting those all together to mean something. Right? Yeah, so association areas are the big part there. So cortex, 3 types of things it can be doing. Each hemisphere, you've got 2 hemispheres, and your body, like, the human body is not perfectly symmetrical, right? Like, people know this, right? Like, the heart's a little bit off-center, your liver's off-center, right? But more or less, you're fairly symmetrical, right? Like, arm, leg, lung, right? All right, line down the middle. If you were Frankenstein and building some sort of monster, right? The obvious solution would be like, okay, if I'm going to have a brain here controlling the body, you'd expect, or at least I would expect, I expect that this part of the brain controls this part of the body. Right? That would be kind of intuitive. It's not. It's not like that. Each hemisphere worries about the contralateral, the opposite side of the body. So if you're right-handed, your left hemisphere is bossing around your right hand. We're okay with this? Vice versa. So contralateral side of the body. The cortex can be lateralized, meaning some cortical function, only happens in one side or the other. This is true, but people take it too far. And there are quizzes online. I have seen them. Quizzes online. There are things like, are you right-brained or left-brained? Pick now. Do you like organization or chaos? And you're like, well, I don't know. Maybe I guess I like organization. They're like, do you like drawing with crayons or typing on the computer? And you're like, I don't know. I guess crayons. They're like, okay, we've decided all about you. They are not scientific, and it's bunk. Okay? That being said, this actually doesn't upset me as much as the snowflake. Well, maybe. Anyways, it upsets me too. What is true is most abilities are living on both sides of the brain, right? Some abilities do seem lateral. So speech typically is lateralized. Not always, but often. Prosody of voice, to being able to not talk in a monotone and actually have it go up and down, that's typically lateralized. There are a couple of things which one side or the other pulls more, but yeah. So it's true, but it's not as true as some people would like you to think. And then conscious behavior, if you're like thinking about stuff and doing stuff, conscious behavior typically involves the whole thing, right? So like, yes, okay, so you've got the cortex. Different areas do different things. Great, this part's doing vision, this part's doing speech. Sometimes between one hemisphere and the other, they do different things, and that does happen, though the hemispheres talk to each other a lot. And if you're doing something complicated like live or read a book or talk to your buddy or make scrambled eggs, you're probably involving the whole cortex in one way or the other. Not every single neuron is on or off all the time, but you're going to pull information from everywhere. Questions, comments? That's where we're going to end on the like, okay, cortex, yay. We'll pick back up on Tuesday about the main chunks. And it's cool, and you get to have like creepy pictures, like this creepy picture here. I don't know, I think it's creepy. Don't freeze on me, computer. Never mind, you'll see it on Tuesday. It's frozen. So I guess we do have to be done. Remember to take a quiz before midnight. Also, good luck on your other exams too, by the way.